Lahjavida, a biotechnology company, aims to transform cancer care by making it safer and more effective. Lahjavida has developed a new approach to treating cancer, a unique tumor-targeting dye that binds to over 30 different cancer types so far has been connected to standard-of-care cancer drugs via a cleavable linker system that cleaves only within the unique environment of a cancer cell. The combination of these components, a tumor-targeting dye, shown in green, an active cancer drug, shown in red, and a cleavable linker system, shown in orange and purple, is called a dye-drug conjugate, or DDC for short. The dye fluoresces, showing doctors where the dye-drug conjugate is located at any given time. One of the conjugates is shown here. The tumor-targeting dye is located at the top, and the cancer drug is at the bottom. In the conjugated form, the cancer drug, now shown in blue, is inactive. The inactive drug is safe, having no effect on cells that it contacts. The cleavable linker system between the dye and the cancer drug dissolves in the presence of an enzyme found in cancer cells, as well as acidic conditions, also found in cancer cells. Both linker segments must be separated from the cancer drug in order for the drug to be activated to kill cancer cells. Here is a representation of an oncology patient with multiple tumors throughout the body, shown here in orange. When this conjugate is injected into the patient, it travels harmlessly throughout the body searching for cancer. In the early hours following injection of the dye-drug conjugate, it accumulates in all tissues, healthy and cancerous. Healthy cells are unable to cleave the linker system and activate the cancer drug. Therefore, the healthy tissues remain safe from its toxic effects. Over the course of 48 to 72 hours, the DDC accumulates preferentially in all cancerous cells. During this time, it becomes less concentrated in healthy tissues. Lahjavida's tumor-targeting dye is taken up at much higher rates by cancer cells, entering the cancer cell via membrane channels that are overexpressed in cancer cells. Lahjavida has been able to create a system that permits the dye to continue to be taken up preferentially by tumors, while being attached to the linker system and cancer drug. Once inside the cancer cell, the linker system undergoes a two-stage cleavage. First, an enzyme, found at much higher levels in cancer cells, attaches to the linker system and cuts it, releasing it from the dye. Then the acidic environment of the cancer cell degrades the remaining portion of the linker system, activating the cancer drug and allowing it to kill the tumor. Once again, this two-stage cleavage mechanism creates an additional layer of protection for healthy tissues from the toxic effects of the cancer drug. Cancer cannot hide from this unique dye molecule. The DDC is small enough to penetrate any location of the body, potentially including the brain. If proven in further studies, this capability would be revolutionary and permit the treatment of previously untreatable cancers. Dozens of different cancer drugs can be efficiently and economically attached to a single dye class. By changing the dye, linker system, and/or cancer drug, the conjugates can be optimized for patients with different cancers. This will enable targeted, more effective cancer treatment for anyone who needs it. Shown here is the image of a mouse shortly following the injection of one of Lahjavida's DDCs. The animal has a tumor grafted onto its right flank. The location of the dye is seen here in various shades of red, orange, and yellow, depending on the concentration of the DDC. Note that the conjugate is initially distributed fairly evenly throughout the body of the mouse. This next image is of the same mouse, taken 4 days later. As you can see, Lahjavida's DDC accumulates very efficiently in the tumor. This image was taken after 9 days post-injection and demonstrates the selectivity of the conjugate for the tumor. The tumor still fluoresces very brightly, showing the presence of the DDC at high levels within the tumor. Note that the intensity of the fluorescent signal in other parts of the body has been greatly diminished over 9 days, as healthy tissues excrete the DDC. These two graphs compare the toxicity, measured in terms of weight loss during the study, of a pure cancer drug, doxorubicin, a common cancer drug used today, and Lahjavida's DDC with doxorubicin as the payload. The first bar, gray, in each graph shows the average weight of the mice at the start of the study. The pure, unconjugated cancer drug is on the left, and the dye-drug conjugate group is on the right. The red bar on the graph to the left shows the weight loss of the group receiving the pure cancer drug. As expected, the pure cancer drug is highly toxic and causes a significant average weight loss of 32%. When we perform the same analysis of the mice receiving a similar dose of Lahjavida's DDCs, we see very little change to the body weight of the mice, 5.5% average weight loss in this case. This data indicates that Lahjavida's DDCs have little to no toxic effects, while the toxicity of the pure cancer drug is significant. This is strong evidence that Lahjavida's DDCs reduce the toxic effects of cancer drugs on healthy tissues. But will the DDCs kill cancer? The graph shown here is the actual tumor size data from the same mouse study discussed previously. The vertical axis indicates tumor size, and the horizontal axis indicates time post-injection. The blue line represents the saline solution control. The red line represents the pure standard of care cancer drug, doxorubicin, behaving as expected, shrinking the tumor. The green line represents Lahjavida's DDC, using doxorubicin as the payload. Lahjavida's DDC shows equivalent tumor-killing efficacy as the pure standard of care cancer drug, doxorubicin. To have such a significant reduction in toxicity while maintaining efficacy is very encouraging. These images demonstrate that Lahjavida's DDCs are not just a dream. Lahjavida's technology has the potential to transform cancer care. We're excited by the data that we've generated thus far and look forward to a conversation with you about how to join us in this mission to enhance and improve outcomes for cancer patients everywhere.